students. So this is culture media part 3 and we are going to continue with our lecture. So bachcho, now let's discuss culture methods. So there is an inoculating loop and inoculating wire. So the inoculating loop and the wire is made up of nichrome and it is used for the inoculation of clinical specimen on a solid or liquid media and to transfer the bacteria growth from one medium to another. And the sterilization is by red hot under the flame. So now the next is the sterile swab. It is used for the collection of specimen for doing lawn culture for antibiotic susceptibility testing, and its sterilization is done by hot air oven. Now we are going to discuss the Muller-Hinton agar. It is an agar which is specifically used for the antimicrobial susceptibility testing. That means for the antibiotics and to detect your zone sizes. So here are the antibiotics, and approximately 6 discs are applied on one plate, and there is a zone of inhibition which is present around the discs, which basically depicts that whether the according to the zone sizes, which depicts that whether the antibiotic is sensitive or resistant. So after applying the antibiotic disc on Muller-Hinton agar, it is incubated in bacteriological incubator at 37 degrees centigrade. So now right now we are discussing about the methods of culture. So culture methods can be of different types. First is the streak culture. Inoculation is done of the specimen is done on a solid media, and it is used for obtaining isolated colonies of a bacteria to perform the test for the identification and antibiotic susceptibility testing, that is AST. So the lawn culture, next is the lawn culture. It is used for AST, bacteriophage typing, preparation of bacterial antigens and vaccines. Solid culture media Muller-Hinton agar is inoculated by a sterile swab soaked in a liquid bacterial suspension. Next is the stroke culture. On agar slope, by streaking the straight wire in a zigzag manner, it is used for biochemical tests like urease and citrate. So next is stab culture. It is done by stabbing a semi-solid media by a straight wire, and it is used for testing the motility, and it is in TSI, that is the triple sugar iron media. Both stroke and stab culture is done. Last but not the least, the pore plate culture. It is used to determine the viable count and quantitative bacteria present per ml of liquid media, that is in the urine culture. Those culture methods were for the solid media or the semi-solid media. Now is the liquid culture. Culture of blood and body fluids which are inoculated directly into the liquid media. Growth is detected by observing the turbidity, and it had advantage for culture of specimens containing small quantity of bacteria and when large yields of bacteria are required, but has disadvantage of getting pure culture from the mixed inoculum, and as such no visible colonies are obtained. One of the method for anaerobic media was Robertson cooked meat media. The other methods for creating an anaerobic environment for the growth of the organism is candle jar. A lighted candle is kept in an airtight container loaded with inoculated culture plates, and the burning candle reduces the oxygen to the point where flame goes off, and it provides the atmosphere of 3.5% of carbon dioxide. It is used for the growth of capnophilic bacteria, that is the carbon dioxide loving, and like Brucella, Streptococcus and Gonococcus. Microaerophilic bacteria like Campylobacter and Helicobacter. The other methods of anaerobic culture methods for the anaerobic culture are Macintosh and Fields jar. Principle, it is the displacement and the combustion of oxygen. Methylene blue is used as an indicator and colorless in anaerobic condition but turns blue in the presence of oxygen. So it acts as an indicator. Next method is enoxomore. Chemical methods, for example, by absorption of oxygen by pyroglycol and chromium and sulfuric acid, and gas spec method. The other methods being the biological methods, which in which the absorption of oxygen occurs and by the incubating uh, aerobic organisms along with the anaerobic bacteria, and it is a slow and ineffective method. The reducing agent in it is thioglycolate broth, and basically the RCM, that is the Robertson cooked meat media. The other is the method is anaerobic chamber, and last but not the least is pre-reduced anaerobic sterilized media, that is also brass. So thank you for listening, and hope you have enjoyed the video. Thank you. Last but not the least, thank you everyone for listening. Thank you.